Hey everybody, so let's take a look at the DNA structure. So let's talk about DNA structure. And when talking about DNA structure, I think it is most useful to start with the structure of a single nucleotide. So DNA is double-stranded and it's made up of base pairs of nucleotides. And so let's start with a single nucleotide. So I'm going to start with a phosphate group here. And so this is a phosphate group. We have 4 oxygens. One of them is double bonded to the P there. And so yeah, that's a phosphate group. Now the phosphate group is going to be attached to a carbon of a 5-carbon sugar. So there's 1 carbon there. I'll put 1 there. I'll put an O here, C here, C, C. Okay, so this is a 5-carbon sugar. And you can see we have this cyclic area right here. This carbon is not in this ring structure. We can number the carbons 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. I don't know why these numbers have these little primes on them, but they do. This is an oxygen, and this right here, this 5-carbon sugar, this is called ribose. We're going to call this ribose. And it's called ribose if there is an OH here and an OH here. So the bond connects the C and this oxygen right there and right here. So in DNA, now this is what the 5-carbon sugar looks like in RNA. So RNA uses ribose as the 5-carbon sugar. DNA uses 2-deoxyribose as the 5-carbon sugar. And in that case, you can see this little 2-deoxy. Well, this is the 2' carbon, right? So this would just be an H right there if this is a nucleotide of DNA. Now, the 1' carbon is attached to a nitrogenous base. And for now, I am just going to write it just like that. And the base is going to be adenine, guanine, cytosine, or thymine. So this is the basic structure of a nucleotide. 3 components: a phosphate group, a 5-carbon sugar, which would be right here, and we'll call this the nitrogenous base, which essentially means it's a compound with some nitrogen in there, a couple of nitrogens. 2 other terms I'd like to bring up at this point. A nucleoside. Now a nucleoside is a lot like a nucleotide, but it just has the 5-carbon sugar and the base. So sugar plus base. Now the nucleotide, that's the sugar plus base plus phosphate group. Now, okay, so this stuff here we should memorize. You should be very familiar with the structure, the general structure of a nucleotide, knowing the base is attached to the 1' carbon. This phosphate group right here, we have attached to the 5' carbon. And so just be familiar with this overall structure, or memorize this overall structure. When it comes to the nucleotides, we are not going to memorize the details of each nucleotide. There are a few things we will need to remember though. So the nucleotides, the nitrogenous bases. So when it comes to the nitrogenous bases, we need to remember a few key things. So nitrogenous bases can be pyrimidines or purines. The pyrimidines have this structure, this 6-atom ring structure right here, where the nitrogens are at this point here and this point here. We're not going to worry about the double bonds, so we'll leave that to organic chemistry or biochemistry. And we have some numbering here, no primes, but we start with this one right here at the base. This nitrogen is number 1, 2. This one's nitrogen is number 3, 4, 5, 6. And the numbering is important because the number 1 nitrogen in a pyrimidine is the one that's connected to the 1' carbon in the 5-carbon sugar. And in DNA, pyrimidines are cytosine and thymine. In RNA molecules have cytosine and some of the nucleotides, but instead of thymine, we see uracil. And so the general structure is something like this, but there are some different side groups that differentiate cytosine from thymine or thymine from uracil. Now, I won't ask you to memorize those side groups, but when we go to look at base pairing in the, I think one of the next videos, we will take a look at how some of those side groups off of this ring allow for the hydrogen bonding between, say, G and C and A and T. Now, purines have a similar structure, but there are 2 rings. So the first ring, I have to start with this one over here. 2 ends, one on top of the other. And this little 4, let me see, 1, 2, 3, I guess 4, 5 atom structure, 5-atom ring structure right here. Looks sort of like this. And this little piece right here, this is, essentially this piece right here of the pyrimidine. So we just need to, if we remember pyrimidine, well, we can drop that in right there. And the numbering is a little different. So this one's number 1, 2, 3, 4, 5, 6, 7, 8, 9. So those are the positions in the purine ring structure. So the number 9 nitrogen is the one that connects to the 5-carbon sugar of the nucleotide. Now the 2 purines in DNA and RNA are either adenine or guanine. And a good way to remember that these are the purines is this memory key, pure as gold, so pure for purine, A for adenine, G for guanine. So as long as you remember pure as gold, you will remember the purines are adenine and guanine. I don't have a memory key for remembering purines have 2 rings and pyrimidines have only 1 ring. That, I think you might need to come up with your own remembering device, or just memorize it. Okay, so DNA is double-stranded with A pairing with T, and A will form 2 hydrogen bonds with T, and we'll look more at the details of this in a future video. In G, forming 3 hydrogen bonds with C. But what I want to do first is, there's a problem, I think it's 1013, and we went over this in class, and it essentially asks us to diagram an AG dinucleotide So a single strand with an A and a G, and then pair that up with a complementary strand, just like you would see it in a double-stranded DNA molecule. So diagram an AG dinucleotide and show it hydrogen bonded to a CT dinucleotide. And by doing this, we'll be able to see that the two strands of DNA are anti-parallel to each other. So one strand will run in the 5' to 3' direction. If one strand runs in the 5' to 3' direction, the other strand must run in the other direction, 3' to 5'. And so by doing this diagram, you'll be able to see where these numbers come from and what we mean by anti-parallel. And let me walk you through how one would diagram this. So I'm going to start with the A's and the AG dinucleotide, and we're not going to worry about the details of the purines in this case. We're just going to diagram the nitrogenous base as a rectangle. And we just want to do this diagram so we can understand where this 5' to 3', how to see these 5' and 3' directions, and how to see the directionality and the anti-parallel nature of the two strands relative to each other in a strand of DNA. So I'm going to start with a phosphate group. And when I'm diagramming a nucleotide, I'm always starting with a phosphate group on the 5' end. And what do I mean? What do I mean when I say 5' end? I mean that 5' carbon of the 5-carbon sugar, right? So if you remember that the 5-carbon sugar, each carbon is numbered. And sometimes when I didn't put the carbons here, I just drew these as angles. We can consider that, okay, I didn't put the C there, but we just need to remember that those are C's. Those are carbons. Why? Well, for one, I numbered them. 1', 2', 3', 4', and I drew this one up here, 5'. So that's a 5' carbon. And again, that's why it's necessary to remember these numbers. Because if you're going to understand the directionality of strands of DNA, you need to understand, how to identify which carbon is 5', which is 3'. So now, like I said, we're not going to diagram the details of the nitrogenous bases. I'm just going to label that one A. So it's attached to the 1' carbon. Now here, this is where we normally have an OH, right? And we'll talk more about how the phosphate diester linkage is formed. But what I'm going to do here is I'm just going to put this O here, and I'm going to connect it to the phosphate here, and another O here, and O here. So now we have the phosphate group. So there was an OH here. That OH is gone. Now we have a phosphate group. And this is an H right here. Right? This is a DNA molecule, so there's no OH for that 2' right here, just an H there. I don't even need to put that in there, it's just distracting. Okay, so that OH is gone. We have our phosphate group here. Now this is connected to the 5' carbon of the next nucleotide. So here's 1', 2', 3', 4', 5'. I numbered the carbons again in the 5-carbon sugar. This is attached to G. And now we have our OH right here. And if we wanted to add another nucleotide here to make a trinucleotide, I would just need to, get rid of this, put another phosphate group here, and then add another 5-carbon sugar with a base. And we can keep diagramming. We can make a very long diagram. Just keep adding nucleotides like that. And that's how DNA polymerase will add nucleotides when it's replicating DNA. It just adds a new nucleotide to this 3' end. So we can see a directionality to this strand. Start at 5' here and ends at 3' here. So 5' end, we have a phosphate. And at the 3' end, we have a hydroxyl group on the 3' carbon. So now the next part of the question was to show another dinucleotide bonded to this dinucleotide here. And in that case, we'd have a C here and a T here, because A bonds with T and G bonds with C. But now I have to put the nucleotides in. I have to do this backwards. So here's the 1' carbon of a 5 carbon sugar, 2', 3', 4'. I'll have this OH here. Remember the OH group attached to the 3' carbon. I'm not putting a phosphate group here because I don't want any another nucleotide down here. I just want a dinucleotide. And this is the end of the dinucleotide. So here's the 5' carbon. I'm attaching this to a phosphate group. Now here's the cytosine of the next nucleotide, and here is the 1' carbon here, two prime, 3', 4', 5' carbon. Let me put a C here, C, 5', and then I can connect this to a phosphate group. Oh, I didn't mean to do double bonds there. I think I did that in lecture today too. I don't know why I like to put double bonds there. Okay, now this carbon right here is connected to that oxygen over there. Okay, so we have our really short fragment of DNA, just 2 base pairs, one here and one here, and you can see the directionality is anti-parallel. So 5' carbon here, we end with a 3' carbon down here, and on this side we start with the 5' carbon, we follow it all the way down, we see the 3' carbon down there. So 5' to 3', 5', 3'. Okay, the strands are anti-parallel to each other. So I hope that helped you see a little bit about or helped you understand a little bit more about the anti-parallel nature of the 2 strands in a DNA double helix. So these guys will wind around each other to form what we call the double helix. And there are some other things we should know about the structure of DNA. And at this point, you might want to pull up the diagram that's in the lecture notes. So some things you'll need to consider or to remember about the structure is that it is double stranded. A pairs with T, G pairs with C. The strands are anti-parallel. There are 0.34 nanometers between base pairs. So let's say if this is A and it's paired with T, and then this is G and it's paired with C, and then this is C and it's paired with G, and A is paired with T again. Now let's say these are the 2 strands, strand 1, strand 2. The distance between these is 0.34 nanometers. And someone in class, after class today, had a good question. What if, is it always 0.34 nanometers? And you know, it's a good question because I haven't really considered if there's tension on the DNA when it's twisting. There's this thing called supercoiling, where if you twist the DNA in a certain way that it causes tension to form, and the DNA will fold in certain ways, and it will form these things called supercoils. And it's one of the things that cells use to help fold DNA. And when you're inducing tension by, say, over twisting a molecule of DNA, does this spacing stay at 0.34 nanometers? And I would say probably not, maybe not, probably goes below that a little bit. So it was a good question. And let's just say that this is, let's say, under no unusual strain, the distance between the stacked base pairs is 0.34 nanometers. Also, there are about 10 base pairs per turn of the DNA molecule. So if you remember in lecture, this is where I went over the diagram with you, and I showed you a nice diagram that's in the lecture notes. Showed you how to look at the diagram and see where one turn is, and there should be about 10 base pairs per turn. I think we now know that to be more accurately 10.4 base pairs per turn. So the original Watson and Crick model suggested 10 base pairs per turn. Later measurements suggested to be 10.4 base pairs per turn. And the distance for one turn of the DNA is 3.4 nanometers. Now, also we know that the DNA is a right-handed double helix. And again, I won't ask you to be able to tell the difference between left-handed and right-handed, but I think we came up with a pretty good method that should allow you to do that should you ever need to in the future. But the Watson-Crick model, DNA is a right-handed helix. It's some more complicated stuff. The form of DNA, this structure right here, refers to the beta form of DNA. And so there's some other forms of DNA that you can get under high salt in other conditions that we won't really talk about. So the B form of DNA is essentially what we're covering. And that's just some extra information if you're curious. So that's it for the structure of DNA. We're going to talk more about hydrogen bonding, those hydrogen bonds that hold the two strands together, in the next video.